Hi guys, today we are going through a lesson on indicators, and this is lesson 5.8. Indicators they're interesting. They're chemicals that change colors. So indicators are commonly used to show approximately where the pH of a substance may be located. So this is a pH paper here that you can use for universal indicators, and so the different colors are associated with different pHs. So green, this is our neutral zone here, and then we get to the very purpley colors, that's our very alkaline, very basic. Acidic is on the red side here. And then we run through our Roy G. Biv here, our rainbow of colors. So indicators are things that change colors. They're weak organic acids. They change color when the hydronium or hydroxide ion concentrations of a solution are changed. So this is an indicator here, an example called phenolphthalein. So phenolphthalein, you guys will do a lab using phenolphthalein. Phenolphthalein is probably the most commonly used indicator, and it turns pink when things start to become neutral. So when you reach the neutral zone, you'll see it's pretty clear through here, and then right at 7 we start getting a very light pink, and then of course we get into the fuchsia zone here in pH 10. So indicators are substances that change color based on the pH of something. Now, so yeah, they change color over a given pH range, and on the back of your Ka tables, you've got a list of sort of common indicators for you to know. And we did talk about these when we did our demo on methyl orange. Remember that orange juice thing? Whatever that demo was. We used methyl orange there, right? So these are just examples. So if, again, phenolphthalein is colorless, and then up until you get to around pH 8 where it turns pink. Litmus, we've got red and blue litmus paper changing depending on the acidity again, and methyl orange then also changes color here as we move from acidic to basic. So those are just some examples. You don't need to copy those down. It's just to show how it works. Okay, so we are actually going to use Le Chatelier's principle to show how indicators work here. So this would be just a general formula, so H indicator. So remember, these are weak organic acids, right? So weak acids means they do not dissociate 100%, so only partially dissociate. And we've got two different forms of it, right? So we've got our acid-base pairs here. So this is our acid here, so this is the acidic form, and this is the basic form here. So acidic and basic forms here of the indicators. Now, how this works is that this will be color 1. So the acidic version of the indicator would be color 1. The basic version is going to be color 2. So color 1, color 2, and this equilibrium will shift back and forth depending on what kind of solution the indicator is put into. So we'll do a question here like this. Whoops. Okay, so again, we've got color 1 and color 2 here. We've got the acidic side and we've got the basic side here. Now what will happen, so what color will show with the presence of an acid? So what will the presence of an acid do to this system at equilibrium here? Now, so you have to think, if you put this indicator in a solution and then you added an acid to it, so made it more acidic, what would happen? So we have to think, if we're adding an acid, what are we adding? So hopefully you have come to the conclusion that we are adding H+. If we add H+, or increasing the concentration of H+, if we increase the concentration of H+, which way is equilibrium going to shift? Yeah, you thought you were done with equilibrium. You're not done. The equilibrium is going to shift to the left, yes. So if we add H+, that's going to increase the concentration of H+. It's going to increase the opportunity for H plus and IN minus to react, driving the reverse reaction. So it's going to shift equilibrium left. And so adding H plus will shift equilibrium to the left, causing a shift from color 2 to color 1. So if equilibrium shifts this way, shifts to the left, then that means you will see color 1, okay? Now, what will the presence of a base do to this equilibrium? So in this particular situation then, if we add a base, excuse me, if we add a base to the system at equilibrium, what's going to happen? Now, what you have to recognize is if we add OH minus and H plus make what? They make water, right? So H plus and OH minus, they want to react together. They are going to react together to make water, which means you are going to be taking away H plus. If we take away H plus, that means we are going to reduce the concentration of H plus because the OH minus is going to react with it. In which case, which way will equilibrium go? Yeah, equilibrium will go to the right, and we will see color number 2 in this situation. So adding OH minus will decrease H plus, causing a shift from color 1 to color 2. So you have to be able to understand how these shifts in equilibrium are changing the color, okay, for these indicators. Now, you're going to work through exercise 39, and it is going to reference quite a bit of equilibrium stuff, so you might have to review that if you're a little foggy on it. But take a look at that and exercise 39. Thanks, everyone.